Thank you, Sohini. Our next speaker is Sally John, who is Vice President of Computational Biology and Genomics at Biogen.
Thank you so much, John. So to Nadim's challenge then, I mean, some of what we've heard this morning sounds like if we could expand our toolbox of drug modalities, then we'd be able to modulate every gene in the genome. But I think there's another side to this as well. To start a drug discovery program, we need a way to assay and modulate the target in cell and model systems in a way that we're confident recapitulates some part of human physiology that we care about. And I think that it's this piece, the translational piece, and better understanding the biology of the targets that is going to make a difference to how we accelerate and translate genomics. So I'm going to buck the trend and talk about some targets that are not human genetic targets. And in fact, in the world of drug discovery, as Lon very elegantly showed, most targets don't have human genetic evidence. And biologists are really very good at coming up with long lists of targets based on some other form of evidence. And the ones we have here are the salt-inducible kinases. They're druggable, and we want to inhibit them. So we've got a head start because the chemists like kinases. And the one we're particularly interested here is SIK2, and this data actually comes from a pan kinase, so a non-selective kinase inhibitor that showed that the anti-inflammatory effects were mediated by SIK2. And SIK2 inhibition has a really desirable effect of attenuating the anti-inflammatory cytokines like TNF at the same time as enhancing anti-inflammatory cytokines like IL-10. And this would be fairly novel in terms of anti-inflammatory medicines. So our team comes to us and says, Sally, is there any genetic data that would associate SIK2 with inflammatory diseases? And this is a slightly messy slide, but in the top left-hand corner here, if you do a PubMed search, you come up with variants in SIK2, which are associated with primary sclerosing cholangitis, which is an autoimmune disease. And you can see the little figure here showing of the 6 loci. This was an ImmunoChip paper, a variant so in SIK2. So this is all there is when you do a PubMed search. We can take this SNP and say, well, what else do we know about it? And if you go to public data like the University of Cambridge MR catalogue, which is run by John Dinesh, you see that the same allele has a protective effect for cardiovascular disease, not very significant, but it's the opposite direction of effect. So is this a potential safety concern? We have a partnership with 23andMe, and we looked at the same SNP in the 23andMe database. And this red line here is an odds ratio of 1. That same variant is associated in the same effect with an increased risk of allergy and eczema, so more immune phenotypes. And again, we see this effect on self-reported blood pressure meds in the opposite direction of effect. Based on what we know about these genes in rodent models, this was a safety concern that was raised by the teams. So because we're now concerned about blood pressure, we looked at all 3 SIK genes in the 23andMe database, and SNPs at some reasonable level of significance have association with blood pressure and blood pressure traits. The middle graphic here is the one from the previous slide, and this is SIK1. We know that the SIK1 knockout mice on a high salt diet has blood pressure, so that's might be expected. Also, we see association for SIK3. Now, we've just joined up to the CTTV, so we had to look at what the CTTV chart said. And this is the same SNP from the 23andMe data that's associated with salt with an iron marker, and that's associated with blood pressure. However, we didn't find this on PubMed because the authors, Thomas Eilig and Kristen Geiger and colleagues from the Helmholtz, attributed this variant to PCSK7, which is actually just a bit downstream down there. These other variants, so sorry, just to orientate you, this is the gene here, and the common variant associations are these little that's here, these are all with triglycerides, and you can see that next gene over is ApoC. So we're now in a world where we have a lot of associations in this gene, but we don't really know whether this is the causal gene. In fact, we expect it's probably not. And this is hugely important to us, because if we want to make a selective SIK2 compound, and we think this is important, we could have a whole army of chemists trying to avoid an effect on SIK1, and it may not matter if this is not the causal gene. So to just summarize all of that data, I think we have provided valuable information for the team. It's not conclusive. One of the biggest challenges is the causal gene of directional effect, and I think almost everyone else has focused on functional coding variants. Given stories like these and experience that many of us have, we also wanted to look at functional coding variants, but not just find new variants that are associated with traits, but ask about potential genes and targets that we're already interested in. And so this grew out of, I think originally it was Nadeem and Robert Plench and myself sat down, and then we got other people on board. And so 4 companies and 3 institutions, Mark from the Broad, the Institute of Medicine in Finland, put together a collaboration really quite quickly. I think we were just talking, within 3 months, we'd got all 7 institutions signed up to a contract to do this. Each company chose some genes. We were aiming for 50. There was quite a lot of overlap. We ended up with 37. And they were a combination of targets in our pipelines that had no previous genetic data and targets with genetic data. And the first thing we've done is an association analysis across all of the traits in the Finnish registries. So this is the same variant that Carrie showed. So as a proof of principle, you see the PCSK9 is clearly protective for myocardial infarction and pretty well all of the other cardiovascular traits. The number of traits we looked at were over 200 quantitative measures and over 169 disease endpoints. And I think one of the reasons that PCSK9 is such a nice poster child is because we do have a biomarker, and we know from epidemiological data, and this is actually from one of Nadeem's publications on the emerging risk factor collaboration, that there is this very clear association between lipid levels and cardiovascular risk. And so it makes a nice triangulation of a DNA variant, a biomarker that we care about, and a clinical endpoint. And in most cases, we don't have the biomarker or the intermediate phenotype that we care about to triangulate that story. One thing that we did find that was, I guess, somewhat unexpected, we looked at prescribed medication, and perhaps not surprisingly, the drug that was most used is that carriers of the variant are much less likely to use lipid modifying agents. However, the second most commonly used drug, and this was an increase, so the ratio is 40% more likely to use psychoelectrics and psychoanalytics. And there were 34 people in this study who were homozygote for this variant, and again, their hospitalization was psychosis and neurosis. So this isn't replicated data, but for us in drug discovery, is this a potential safety indication? I don't know whether these types of drugs and indications would overlap with the neurocognitive effects that we are seeing occasionally on PCSK9 inhibition. But this is the sort of data that we would pay attention to. So just to finish off then, I've talked through 2 examples today. I hope, I have highlighted that a lot of what we do is still targets without genetic data, and we are looking as much as possible for summary data to try and better understand what the world of genetic data looks like. So for the SIK2 example, we have a gene and we're looking across multiple resources to try and better understand the genetic information. And clearly in Finland, we've got one resource and we're looking at multiple genes. I think the more that we can bring those two worlds together to get a complete picture, the more able we are to do this in a timely fashion. And it's time that is really critical for us. If we start a drug discovery program, we start assays, we start high throughput screening, we're on a road that's going. So we need to be making decisions fairly quickly in the life cycle of a project. And I'm going to leave it there, and Meg is going to continue some of the discussion around this pre-competitive effort that we've been discussing this afternoon. Thank you.